Differential stains are important tools in the microbiology lab. In this video, I'd like to introduce you to these stains and talk about why they're important. Bacteria are colorless cells. I know. By now you've seen red or yellow colonies, maybe even pink ones. That color is from a pigment that the bacteria make and then secrete into their environment. The cells themselves have no color. This means that if we want to look at bacteria under a microscope, we need to add color. In the microlab, we use biological stains for this purpose. These stains are composed of a solvent, which is usually benzene, a chromophore, which is a chemical that imparts color, and an ionizing agent called an auxochrome. That's AUXOCHROME. The auxochrome allows the chromophore to form a salt with the charged components of a cell. This imparts color. The important thing here is that the stain becomes chemically bonded to the cell, and a simple rinse with water is not enough to get rid of that color. Here's an example. This is methylene blue. The chromophore ionizes this stain so that we lose a chloride ion, and the chromophore here, this whole chemical, becomes positively charged. Now, the surface of a bacterial cell is negatively charged. So when we add this stain to bacteria, the positively charged stain is attracted to the negatively charged surface of the bacterium, and the two combine, forming a salt. This stains the bacteria blue. And with this type of stain, we can get a little bit of information about the bacteria. This stain is called a simple stain because it uses only one stain, which is the methylene blue, and with it we can see information like the size, the shape, and the arrangement of bacterial cells. Differential stains use more than one stain to color cells or the structures of cells. So differential stains use more than one stain to distinguish among cells based upon their structure. Now notice that here we have a differential stain. We can still see the size, the shape, and the arrangement of the cells, just like with the simple stain. But the differential stain gives us a little information about the structure of the cells, in this case, the cell wall. This is a Gram stain. So we know that Gram-positive cells stain purple, Gram-negative cells stain pink, and that's tied to the structure of the cell wall. So when we do a differential stain, there are four chemicals that are involved. The first stain is called the primary stain, and it's going to stain everything on the slide one color. The second chemical is a mordant, and I must say that the mordant isn't always a chemical. Sometimes the mordant is heat or steam. But the job of the mordant is to increase the attraction or the affinity of the primary stain for cellular structures. The third component or chemical is a decolorizer. The decolorizer selectively removes the primary stain from cells or specific cellular structures. The last component is a counterstain. So counterstains stain decolorized cells a contrasting color to the primary stain, so we can distinguish those structures that were decolorized from those structures that weren't. So here are some examples of differential stains. The Gram stain is a differential stain. It distinguishes cells based upon differences in their cell wall structure. The acid-fast stain is a differential stain because it distinguishes between cells that have mycolic acid and cells that don't have mycolic acid. The endospore stain, which we won't really cover in this class, distinguishes cells based upon their ability to make endospores or not make endospores. And then the capsule stain distinguishes cells based upon their ability to make a thick, well-ordered capsule. To give you an idea of how a typical differential stain works, let's look at the Gram stain. So the first step of a Gram stain is to add the primary stain, which is Gram's crystal violet. And Gram's crystal violet stains everything on the cell purple. Next, we add our mordant, which is Gram's iodine. So remember, Gram's iodine binds the crystal violet, making a crystal, a large particle. And that is held onto by the peptidoglycan a little bit better than just the crystal violet itself. But still at this step, everything is purple. Next, we add the decolorizer, which is alcohol. Now, when we add alcohol, remember it dehydrates the peptidoglycan. And as a result, for your gram-positive cells, because there's so much peptidoglycan there, the Gram's crystal violet iodine particles become trapped within the cell, basically within the cytoplasm. Some places that you read will say that the particles become attached to the cell wall. I like to think of it as that the particles are being trapped. For your gram-negative cells, on the other hand, remember that alcohol can remove lipids from a cell. So the outer membrane gets dissolved and washed away. When the peptidoglycan layer gets dehydrated, it develops rips and tears, which allows the crystal violet particles to be rinsed out. Lastly, we have the counterstain. So after decolorization, our gram-positive cells are purple, but our gram-negatives are colorless. So we add safranin to add some color to the decolorized cells. The gram-positive cells have so much stain within them already that they can't really absorb the safranin, and what little safranin does get absorbed is usually overpowered by the purple, so you don't see it. So that just leaves the gram-negative cells to be stained. So now we've been over the difference between your simple and your differential stains. Remember, simple stains, everything is stained one color because we're only using one stain, and we get size, shape, and arrangement information. Differential stains use more than one stain, and we get size, shape, and arrangement, but also structural information. Remember that there are 4 basic steps or chemicals to your differential stain. You have a primary stain, a mordant, a decolorizer, and a counter stain. You'll want to know that information for your lab exam, lab quiz. Your differential stains are important diagnostic tools that guide doctors in treating disease. And now that we've been over the basics, you're ready to learn more. Your next step is to review the videos on the specific differential stains as they become available. Until then, keep studying and keep learning.